glycolysis. So let's talk about glycolysis first. What does that word glycolysis sound like to you? We've also heard some of those words before. They make it up. Yeah, breaking of something, right? Lysis, like rupturing or breaking or separating. And glyco, I don't know if it sounds like this to you, but it sounds like sugar to me, like glucose or something like that. So the process of glycolysis is literally the splitting of sugar. And so what happens in glycolysis is we take one glucose, which I'm going to illustrate with little black blobs to indicate each carbon. All right, I can't think of a better way. So if you imagine, here's glucose. There's six carbons all connected together into a chain. Right? In glycolysis, we're going to split that one glucose into two molecules that are called pyruvate. And those two molecules are going to be three carbon molecules. So we basically broke one of the bonds. Do you want to see what actually happens, what's involved in getting from glucose to pyruvate? This is glycolysis. All right? You take glucose, hexokinase adds a phosphate. Phosphoglucose isomerase isomerizes it. Blah, 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 blah. 9 steps, 9 reactions getting you to pyruvate. Many non-majors textbooks literally go through every single step of glycolysis. Why? I think to obfuscate the main idea, as far as I can tell. So what we're going to do, and in case you think I'm unfairly burdening you with detail, I show this because we're summarizing this entire slide with the word glycolysis. All right? And all I need you to remember is you go from one 6-carbon to 2 3-carbon molecules. All right? Now the other thing that I want you to notice is that you actually have to spend a little energy to get a little energy. So you put in 2 ATPs, and then for each of the 3 carbon molecules, you harvest a couple ATPs. So our net balance, we're coming out with a teeny little drip of ATP, but we haven't risen to the category of lots of ATP yet. There's just a teeny bit that's been taken. And glycolysis, the reaction occurs here in the cytoplasm of the cell. I show you this slide to get you on my side. It's like a secret trick to show you how bad I could have made it, and then you think that how bad I have made it isn't that bad. I know I'm not supposed to reveal that, but you guys probably know that anyways. So we then move on to our next step, which is called pyruvate oxidation. And in pyruvate oxidation, we chip off 1 carbon from each of these 3 carbon molecules. All right? And I'm going to illustrate that up here. We've got our 2 pyruvates. Let me just stick with my... We chip off a carbon from each, and we generate these molecules that are called, anybody know? I wouldn't expect you to. Starts with an A. C-E-T-Y-L.
Acetyl-CoA.
Acetyl-CoA. So, if you are keeping score at home and you like terminology, this word refers to a 2-carbon molecule. And the CoA refers to something called coenzyme A. It's just a different molecule that has to be present for the enzyme to act. And I'm going to show you it in a second, and it will all become crystal clear. All right? So sticking with this structure, we've got our 2 pyruvates. We're going to chip off 1 carbon each to generate 2 acetyl-CoAs plus 2 CO2s. So I don't know if this helps or hinders, but 1 6-carbon gets split into 2 3-carbons. 2 3-carbons get split into 2 2-carbons, and 2 CO2s get released. And these are the reactions of pyruvate oxidation. It's also kind of complicated, right? So we're going to abbreviate it as pyruvate oxidation. All right? And you do make a little bit of the NADH, but not much of it. Okay? How are you guys doing? So if you look back at our formula at the top, how much of it have we accomplished so far? Formula up here, that is. Have we consumed oxygen?
Yes.
Where?
Well, we consumed it. Okay.
We haven't yet, right? We have not talked about oxygen. Have we made water? No. Have we completely broken down glucose? Not completely. It still exists as 2 2-carbon molecules, but we've released 2 of the CO2s, and we've made a little bit of ATP and a couple molecules of the NADH. Okay? Everybody good at that? So then we move on to the citric acid cycle, or the Krebs cycle. And in the Krebs cycle, what we do is we feed in the acetyl-CoAs, and we completely break them down, releasing all the carbons as carbon dioxide. Or we release 2 carbons as carbon dioxide per acetyl-CoA coming in. All right? So in the Krebs cycle, what we do is we take our 2 acetyl-CoAs. There you go. And we release 4 CO2s. Yeah?
Real quick, doesn't Krebs have an S on the end?
Krebs, yes. Thank you. So each one of the carbons that comes in has been released now as a carbon dioxide. Or at least 6 have been released for every 6 that have been inputted.
What do you mean by oxygen coming in then? I assume that when you say take in oxygen, you must have oxygen in order to release on either step CO2. Like, in order to release carbon, you have to intake oxygen. So both steps require oxygen.
What I mean by use up oxygen is, have we seen any chemical reaction that occurred yet that required oxygen to be present?
Isn't releasing CO2 a chemical reaction?
Yes.
Okay.
So I'm not sure.
Like, then we took in oxygen to release a chemical reaction, CO2.
We haven't. So when you breathe in molecular oxygen, there's a particular place in the whole process where that oxygen gets utilized. All right. So the molecular oxygen that you're intaking, it has a particular utility to the cycle. And so there is oxygen that's being released with the CO2, all right? But it's not an oxygen, like an oxygen gas that's being connected to the carbon and just boiled off. All right, there's a particular place where the actual oxygen you inhale fits into the process. All right, we'll see exactly where that oxygen comes in. So the other thing that occurs during the Krebs cycle is that we make, in addition to these 4 carbon dioxides, we also make several NADHs.